Okay, this is our second and last segment on the extracellular matrix. So what we're going to look at here are some proteins that bind the extracellular matrix to cells. So we're going to look at primarily the fibronectin molecule, which you find in the extracellular matrix, and integrin proteins. And I think that if you're going to focus on one of these and not the other, then integrins are more important. But hopefully you've got time, you can focus on both. Okay, so fibronectin, it binds cells to the extracellular matrix and it helps guide cell movement and really influences cell motility. So this is one of a family of adhesive glycoproteins that form kind of direct links between the extracellular matrix and the plasma membrane. This family of proteins, including fibronectin, have multiple domains to bind molecules in the extracellular matrix, as well as receptors on membranes. And so we'll take a look at the structure here. These are, the fibronectins are glycoproteins, so they have sugars added to them. And what happens is that the RNA that gets transcribed for the fibronectin family of genes undergoes alternative splicing. I hope you remember about alternative splicing. That's where you move different sets of introns to stitch together different exons to give different amino acid sequences, and so you can produce kind of different domains in proteins. Eddie is hunting flies, that's what he's doing right now. And so you get many variants of the protein to give you the ability to bind lots of different types of extracellular matrix molecules. So what we have here are two large protein subunits, and they're attached down here at the carboxy terminus by a pair of disulfide bonds. Each of the fibronectin subunits is folded into this series of kind of rod-like domains, and several of these domains bind to one, maybe more extracellular macromolecules. That includes things like collagen, heparin, and fibrin. Other domains bind cell surface receptors via a short sequence of amino acids here called the Rgd motif. Thanks, buddy, that's lovely. Oh, thank you, give him daddy kisses. So this Rgd sequence here is a sequence of 3 amino acids: arginine, glycine, aspartic acid. And so that can be used to bind certain cell surface receptors, and amongst that will be the integrins, which we'll take a look at in a moment. So this fibronectin acts as a bridging molecule between cells and the extracellular matrix. Give you an idea of how important this molecule is. If you take mice and you delete their fibronectin genes, they have severe defects in their musculature and in their vasculature. Furthermore, many types of metastatic cancer cells, those are cancer cells which spread, those are the really bad, dangerous types, they don't produce fibronectin, so they aren't they're not having their motility regulated in the way that most cells would. You also find a soluble form of fibronectin in blood plasma called plasma fibronectin, and its basic function is to basically drive blood clotting because it has domains for binding fibrin, and it can bind to blood platelets, and so it attaches the blood platelets to fibrin as a clot forms. And so you can see that the protein also forms these fibrils in tissue, and so it's forming these long strands to regulate the cell's behavior in the extracellular matrix. Integrins also bind extracellular matrix, and they're also involved in signaling. So we'll come back to that a little bit later. So these integrins are a large family of cell surface stress receptors that bind to fibronectin or proteins, other kinds of proteins, laminins, for example. And so these integrate the cytoskeleton. So there'll be a cytoskeleton binding region on the cytosolic face of the membrane, and then this sequence up here, or this part of the protein up here, will bind to the extracellular matrix, for example, fibronectin binding via the Rgd motif we just mentioned. So an integrin has got two large transmembrane polypeptides, an alpha subunit and a beta subunit. And so integrins vary. There's a large family, so the integrins vary. They differ from one another in their binding specificity, so the molecules to which they can bind in the extracellular matrix, and the sizes of the different subunits. So the extracellular parts of the alpha and the beta subunits form the binding sites of the integrin, and so the specificity here for different components in the extracellular matrix is largely due to the alpha subunit up here. So there are multiple types of alpha and beta, and so you can have alpha 3, beta 6, all different kinds of combinations of alpha and beta subunits, and that gives you many different combinations of the heterodimer, because you can put lots of different types of alpha with lots of different types of beta. So that gives you a lot of diversity in the types of integrin that you can produce. So that's kind of a little bit about integrins. Let's take a look at more specifically how they connect the extracellular matrix to the cytoskeleton. So the tails of the integrin interact with cytosolic proteins. So here's the tail of the integrins down here. Here are the integrins, this purple and this blue structure. And so the tails, the cytosolic tails of the integrin associate with cytosolic proteins which link to the cytoskeleton, and here we're linking to an actin filament, so a microfilament. Excuse me, I have an itchy knee. Now, if you're a migratory or a non-epithelial cell, basically, you use these integrins to form these focal adhesions, which are here in purple. These purple dots are focal adhesion contacts, and so that's where you get these clustering of integrins, and that can allow the cell to be migratory. So there's going to be forming the focal adhesion, then breaking it, and then forming and breaking as the cell crawls through, for example, the extracellular matrix, if you're a fibroblast. And so you get these clusters of these linker proteins and integrins in these focal adhesions. So things like vinculin and tallin and alpha-actinin, they're all in these focal adhesions to link the cytosolic tails of the integrins to the cytoskeleton. So those are focal adhesions that you find in migratory kind of cells. In epithelial cells, we're going to find integrins clustered into structures called hemidesmosomes. Think back to one of those early slides, and it had the epithelial cell with the microvilli on top, and on the bottom there were hemidesmosomes, and the desmosomes were on the sides. Those were the cell adhesion contacts. The hemidesmosomes were on the bottom, attaching that cell to the extracellular matrix. And so in our hemidesmosomes that you find in epithelial cells, we're going to find things like alpha 6, beta 4 integrins. Don't worry about the specifics here. And in this case, the integrins will be attached to keratins in the extracellular matrix. Who's this one? Oh, this is Henry. Henry usually doesn't appear, so this is Henry making an appearance. You can always tell Henry because he's got a giant head. Sorry, the integrins are attached to keratin in this case here. So the keratin would be in the extracellular matrix, and the integrins are attached to the keratin, anchoring the keratin to the integrin to the cytosolic intermediate filaments. In this case, that's the cytoskeleton that's being used in this example. Okay, buddy, off you go. So the linker proteins here form these really dense plaques, which then the intermediate filaments attach to. I think that's all we really need to worry about in terms of integrins and fibronectin. We'll leave it there, and I'll see you soon for our next chapter. Bye for now.